And we're moving on to molecular complexes. And we're going to start in the nucleus, where you have chromosomes. Human DNA, even though it's a molecule, if you could unspool it, would be about that long. But it needs to fit into a fairly small room, so there's a whole bunch of proteins that it literally spools on. We'll talk about these later. But the interactions of the histone proteins in the DNA makes it a molecular complex. And it's a complicated molecular complex because you need access to individual genes when you need to make individual proteins. So there's a whole bunch of stuff. There's a matrix in here that's going to connect us. One of the things we're going to talk about pretty soon, the cytoskeleton. Now this blob, sometimes there's one, sometimes there's more than one. Inside the nucleus is also a different type of molecular complex. So. Don't blame me, I didn't name it. Why don't we take a common word, a structural term, and just let's put an extra L in it. That won't be confusing at all. A nucleolus or a nucleolus, proper Latin pronunciation, it's a nucleolus. A lot of people pronounce it nucleolus because they didn't take a couple of years Latin. It's not easy to do anymore. When I was in middle school, we had Latin. And incidentally, if you're curious, and I may have already said this, you come three in from the syllables from the end and stress that one. And that's typically for long Latin words where the stress is. The nucleolus is RNA does a lot of stuff. We talked about RNA when we talked about nucleic acids. And it does a tremendous amount of stuff involving genetic expression. So you need processors for RNA. And that's what the nucleolus is. It stores a lot of the components so that you can build it when you need to build it. There's a bunch of different RNAs in there. Particular parts of chromosomes kind of thread through the nucleolus and interact with it in different ways, but so there's particular interactions between the chromosomes and the nucleolus. As I said, how many of them are in there does vary from cell to cell, varies from species to species cells, and sometimes they're visible with our light microscope, sometimes they're not. Ribosomes, where proteins get made, are complexes of RNA, a few different types of RNA, and proteins in this structure that kind of sort of looks like an acorn with a cap and a kind of a round bottom. And the rim of the cap, it does a lot of the processing. Messenger RNA, we'll talk about this later. Messenger RNA from the nucleus goes out and basically skirts along the ribosome. As it goes along, little RNAs called transfer RNAs come in and connect a codon at a time and bring in an amino acid to attach to a growing string. So as the long string feeds through, little ones come in and clip amino acid and it moves, clip amino acid and it moves, clip amino acid and it moves. And you're building the protein as this long strand of messenger RNA feeds through. As I said, we'll talk about this in more detail later. That was just a quick introduction. I'll ask you questions about it when we actually hit it. And then you got MTOCs. So they pulled the T out of microtubules to give this initials. I'm trying to think whether we're as bad at putting together things that are just initials as some other fields like computer science. It's terrible. Chemistry doesn't tend to do it that much. There'll be some things where you'll have to recognize the abbreviation. I mean, DNA and RNA, it's pretty, things that are, long named chemicals very often are initials. These guys build the microtubules that we'll talk about in just a minute that are part of the cytoskeleton and do other specialized jobs. But where these are built, they generate microtubules. You have microtubules that go up cilia and flagella. And what you've got at the base is building the microtubules that are going to move these things. And those are called basal bodies. In a dividing cell, there's a centriole embedded in a centrosome that's an MTOC, and it generates the spindles, which are microtubules. And there's other reasons that there's microtubules in cells, and these also require basically a place to build the microtubules, to stack the tubulin to make the tubules. And that is all part of the cytoskeleton. Which has three potential components. So they discovered these little tubules. Well, these little tubes. Anyway, what are we going to call them? Remember, we're starting to integrate a lot of English in. Well, micro is really easy to borrow because it means little, and people are used to micro meaning little. And so they'll call them tubules, microtubules. And then they found components that are long, thin strings. Took a while because they're smaller than the microtubules, but they're not hollow. They're not like a long straw. And they call them, we'll call them microfilaments. And then they found thicker versions of those that are not hollow, but are not as thin as microfilaments. And it's like, oh crap, we've got something in between. What are we going to call that? So they came up with the term intermediate filaments. All of these are involved in structure and movement. There's two things about microtubules I want you to remember. Microtubules can interact with each other. They pull on each other and make things spin or wave or move or pull in various ways. But inside the cell, they also act as conveyor belts. So you'll have a microtubule anchored over here and it's anchored over there, and you can clip things onto it that will travel along the microtubule. One of my old professors was studying radiolarians. Radiolarians have very long, thin pseudopods that help them to float. We'll talk about that before the end of the chapter here. But they also snare food objects and take them in at the end of these very long, thin projections. And under the microscope, you can see sometimes materials are moving out these pseudopods in order to do stuff out at the end, and sometimes food particles are moving in. And under a light microscope, they make a bump, a traveling bump. And what you see, he showed us the films, is a bump comes in from the outside and up from the inside, going up the middle. When it gets near each other, they go around each other and keep going. He was one of the early researchers on electron microscopes. And before they were able to get an electron micrograph of what was going on in there, it's like, how the hell are they doing this? Because mostly what looks like is happening inside a cell is flow. And it's hard to explain that as flow. But it turns out that those pseudopods are packed full of microtubules. And one material is clipped onto Dynein, which pulls in one direction, moves it across the microtubule in the other one direction. One of them is clipped to Kinesin, which you clip onto the microtubule and it goes the other way. So you can use microtubules for like a railroad track. You can go in either direction depending on which way the engine is pointed. I know that's not entirely true for trains, but you get the basic idea. So Dynein and Kinesin are basically the little clip roll things that you can clip a material, a molecule onto the Dynein and away it goes, or clip it onto Kinesin and away it goes. And move stuff around pretty rapidly inside a cell. Endoplasmic reticulum's not real quick. Stuff is moving through the liquid of the endoplasmic reticulum. Microtubules are fast. Put something on there and it zips to where it's going. And sometimes that's what you need. Microfilaments are primarily made of actin. We've talked about this before. Actin is one of the interacting proteins in muscle, actin and myosin, but actin is also very involved in moving stuff, and also cables of actin are very much involved in giving structure to things in cells. Most of these, in us we've got the skeleton and we got the muscles doing kind of different interacting things. Here, a lot of the skeletal elements are also capable of producing movement. And then intermediate filaments are kind of weird. There's a whole bunch of different classes of them. There's actually 2 class designations because people have worked in different areas, and one of them designate classes by the types of cells you find them in. One of them designates them by the material that the microfilament is made out of. There could be 6 of them, 6 classes, there could be 9 classes. It's a whole thing. These often appear in cells that undergo significant what's called mechanical or physical stress, where it looks like what they're doing is reinforcing the cell to try to minimize damage. So you don't always see them in cells. Even cells that make them, they're not always there. But if you stress the cells, especially physically stress the cells in certain ways, then suddenly there'll be intermediate filaments, kind of like, let's hang on, don't let the cell rip. Okay, I know I never do this right, but I am trying to make these videos shorter. So that's a good stopping point, and this is a good stopping point for the video. The video for the end of the chapter, hopefully it'll be about this length.